Dear viewers, greetings. This present video is about an introduction to the Archaea. Archaea is prokaryotic in nature, and the word Archaea is derived from the Greek word archaeos, which means ancient or primitive, and indeed some Archaea exhibits the characteristics worthy of that name. The Archaea is unicellular in nature, and it is measured in micrometer, and its size was ranged from 0.1 micrometer to 15 micrometer. And in 1977, Carl Woese and George Forts recognized that the archaea have a separate line of evolutionary descent from the bacteria in phylogenetic tree, that is based on the sequencing of 16S ribosomal RNA or rRNA, and he divides the prokaryotes into two evolutionary domains. They are bacteria and archaea as a part of three domain system. And next, the shape of the archaea is spherical, rod-like shape, spiral, plates, irregularly shaped, lobed, needle-like filamentous, rectangular rods, and flat square shape are the various shapes of archaea. And the cell wall of the archaea is made up of polymers other than the peptidoglycan. Because of this reason, archaea was differentiated from the bacteria by Carl Woese and George Forts. Therefore, the absence or presence of peptidoglycan is a distinguishing feature between the archaea and the bacteria. And regarding the reproduction, asexual reproduction is the only way of archaea. They reproduce asexually in binary fission, budding, or fragmentation. And next, no archaeal member has been reported to undergo the endospore formation. Regarding the locomotion of archaea, some archaea are motile due to the presence of flagella and few are non-motile. Regarding the pathogenicity, the archaea are non-pathogenic in nature. Next is members of archaea. Members of archaea includes four important members. One is extreme thermophiles, second one is extreme acidophiles, the third one is extreme halophiles, and the fourth one is methanogens. Extreme thermophiles lives in high temperature. For example, Pyrolobus fumari is a species which holds the upper temperature limit for life at 113 degrees Celsius and found in hydrothermal vents. The second one is the extreme acidophiles. This extreme acidophiles lives in high acidic condition. For example, the species of Picrophilus, which were isolated from the acidic soils in Japan, are the most acid tolerant organisms, and they are capable of growth at around pH 0. The third member is extreme halophiles, and this extreme halophiles lives in high salt concentration. For example, the genus Halobacterium survive in hypersaline environments such as salt lakes and can outcompete bacterial counterparts at salinities greater than 20%. And the fourth and final member of archaea is methanogens, and these methanogens are used for biogas production and the sewage treatment. And these methanogens produce methane gas as a metabolite or metabolic byproduct and are found in anaerobic environments such as in marshes, hot springs, and the guts of the animals, which includes the humans. Next is habitat of archaea. Archaea were originally discovered and described in extreme environments such as hydrothermal vents and terrestrial hot springs. And archaea were also found in a diverse range of highly saline, acidic, and anaerobic environments. Archaeal groups inhabiting different extreme habits, and they are first one is halophiles. Halophiles lives in extreme salt conditions like salt lakes and brackish waters. The example for halophiles includes Halobacterium species. The second one is the thermophiles. The thermophiles lives in extremely high temperature like hot spring and vents. And the example for thermophiles is Methanophyros candleri. The third habitat is alkaliphiles. This alkalophiles lives in extreme alkaline conditions like marine hydrothermal systems. An example for this alkalophiles includes Thermococcus alkaliphilis, that is which was widely regarded as marine archaea. The fourth one is acidophiles. The acidophiles lives in extremely acidic conditions like dry hot soil and volcanic sites. The example for the acidophiles is Picrophilus toides. Mode of nutrition in archaea. Based on their preferences of source for deriving energy, the archaea are divided into different nutritional groups such as phototrophic archaea, lithotrophic archaea, and organotrophic archaea. First one is phototropic archaea. Some species of archaea are known to utilize energy from the sun. Hence, they are called as phototropic archaea. Although they can utilize sunlight like plants, they cannot fix atmospheric carbon. So, they are phototrophic and non-photosynthetic. Example of this phototrophic archaea includes Haloarchaea and Halobacterium species. The second mode of nutrition is lithotrophic archaea. Some species of archaea are known to utilize inorganic compounds or the chemical energy to take care of their energy needs like metal ions, hydrogen and ammonium, etc. They are referred as lithotrophic archaea. The examples for this lithotrophic archaea includes Pyrolobus, Ferroglobus, Methanobacteria, ammonia oxidizing archaea, and sulfate reducing archaea. And the third mode of nutrition in archaea is organotropic archaea. Some of the archaea are known to utilize organic compounds to take care of their energy needs like pyruvate, starch, maltose, etc. And these kind of archaea are referred as organotropic archaea. The examples of organotropic archaea includes methanosacchinale species, pyrococcus species, and sulfolobus species. Finally, association between archaea and eukaryotes. Archaea are also found living in association with various eukaryotes. For example, methanogenic archaea are present in the digestive system of some animals, which includes humans. Some archaea form symbiotic relationship with the marine sponges. This both are the relationship or association between an archaea and eukaryotes. Dear viewers, thank you for your support. Thank you.